Okay, perfect. Our second speaker today is Emmanuel Noutahi. He will be introducing all of you to molecular representation and scoring, expanding on some of the stuff that Barak have just discussed earlier. Emmanuel is a principal scientist and the lead of the Biomodality Research Unit at Valence Lab, which is a research engine within Recursion Pharmaceutical. His research spans representation learning, generative modeling, and human-in-the-loop approaches, primarily aimed at accelerating and redefining scientific discovery. Emmanuel is also actively involved in many open source initiatives, furthering the advancement of ML application to the problem of drug discovery. He'll be presenting some of those approaches very soon. Thanks again, Emmanuel, for accepting our invite and presenting your work here today.
Thanks, Prudential. I'm very excited to be here today. I think that part of my presentation has been covered by Bharat already, so there will be a lot of repetition, but I hope you don't really mind about that. So let's start about first, like, why do we use computation or machine learning in drug discovery? So this is a pretty fun graph. I'm pretty sure that some of you knows already about like the Moore's law, where basically compute is getting like faster and cheaper over time. But in reverse, what we are seeing is that drug discovery is getting more expensive, and we are actually kind of like putting a lot less drug out during the last like decade and so. So like, why are we seeing that? Why are we kind of like getting cheaper compute, but we are struggling to get more drug out? So you might think about maybe that's because we currently have like a more constraint about how to actually bring a drug out on the market. But some people might actually see an opportunity there. Maybe we can use computation, maybe we can use machine learning to kind of accelerate and reverse what we are seeing about bringing a new drug on the market over time. So that's basically where machine learning is trying to have like an impact right now. So like, can we reverse this process, or at least kind of like try to like balance it out a bit? And you have probably also seen a lot more about the drug discovery funnel, where you start with like a lot of potential drug candidates, and then going through the cycle of drug discovery, you kind of like you are losing out a lot on the number of like candidates that you're bringing from compound on the preclinical stage to compound to clinics, compound that you can actually like market as drugs. And the kind of like key hypothesis of like machine learning for discovery is, can we actually change that funnel? Can we kind of impact it in such a way that we can not only increase the number of hypotheses that we are making when it comes to like what we think about biology, what we think about the kind of like the relationship between the structure of a compound and its biological effects, but also can we accelerate that process? So can we reduce it from several decades to bring a new drug on the market to like maybe just a few years, or potentially like a few weeks? So that's kind of like the key like impact that machine learning would like to have when it comes to like the drug discovery. So these are things like people working at the intersection of like machine learning and drug discovery are working towards. So like accelerating the discovery process, but also like increasing the number of like candidates, the number of hypotheses that we can test quickly in a short amount of time. Okay, so you have also probably like seen kind of like this cycle of drug discovery where you go from making some biological hypothesis, like having some insight about which target you want to drug and why is it relevant to a specific disease, and then finding some compound that will be able to modulate some activity of that target, so either inhibition, activation, and such. And then moving from there to like actually finding compounds that have nice properties, so not just activity, but also like drug-likeness, so compounds that are not toxic, compounds that have like good metabolism, compounds that have good solubility, permeability, and such. So those are kind of like constraints based on the drug discovery program that you're running. And then from there, like you run through some in vitro experiments and potentially some in vivo experiment, and then you want to bring that into clinic. So you want to test, run like clinical trials, like test that on humans. And also there, like you need to make sure that even though you have compound with potentially nice kind of like property overall, that they still have the efficacy that we need when it comes to like human trials. So it needs to like take into account things like formulation, dose, and such. So there are other parameters that you need to kind of like account for during all that process. So it's kind of like a very long cycle. And the goal with like at least like scoring when it comes to machine learning is to find things where rather than having to wait all the time to run expensive experiment and then get some feedback, can we have a proxy model that will tell us how good a compound is for a given parameters? And if we're able to do that, then we can screen a larger amount of compound because in vitro and in vivo experiments are very costly, and you cannot just like take a random compound and test that on human. It's not ethical, and also like I don't think like no one would like to let you like test a random compound in their body. So this is where like there is some opportunity to think about how we can build machine learning model that will take a compound and will be able to predict the activity of the compound or some given properties like solubility of the compound. Cool. So first question is that what do we actually score when we are trying to build like scoring function for drug discovery? So you can think about the binding affinity of a small molecule to the target. You can think about like any type of like a potency assay that you have that measure like the activity of the compound. So it does not need to be binding. It can be like functional assay, it can be like cell-based assay and such. We can think about like trying maybe to figure out what's the mechanism of action of that compound. So does it like inhibit the target? Is it like activation and such? So you would like to be able to predict that. But you also care about things like selectivity. So is my compound only having an effect to the given target of interest, or is it actually binding to anything and kind of like interacting with all the protein that you might have in the body? You care also about things like absorption, distribution, metabolism. So how stable is the compound in the body? How quickly is the body able to like to remove the compound from your body? So if the compound is not able to stay that long in your body, that's also an issue. You think about things like permeability. So is your compound, is it able to like to pass through the cellular membrane or not? So because if not, cannot get into the cell, that's an issue. And when you think about permeability, you also need to think about efflux. So efflux is kind of like active transportation of a foreign element outside of the cell. So you have things like transporter protein that can pump out a given foreign element out from the cell. So you want to predict that. So even if your compound is able to enter the cell, if there's a mechanism in place that remove it from the cell, it's still not good enough. So things about like synthesizability. So like if you do have a compound, but if you actually don't know how to make it, it doesn't really matter because you cannot produce that compound, you cannot test it and such. So like there are a lot of like kind of like scoring parameters that you need to take into account whenever you think about drug discovery. And historically, like people have been doing that with like food trials and errors. So like you have some hypotheses about your compound, about the structure of the compound and the impact of that structure on some activity or some property of the compound. Then you test in the lab and then you go back and then you have some feedback that you can use to improve the design of your compound and such and such. So can we actually use machine learning to make that faster and to scale it in such a way that you have maybe 1 billion of compounds and then you have a set of models that will tell you for this compound, here are the parameters that we can predict about them and how certain we are about those parameters. Okay, so typically when you try to apply machine learning in a drug discovery program, it looks like something like this. So you start with data that are maybe specific to your project. So it can be like you run a high throughput screening, and then you get some kind of like a data bag that tells you whether the compound has the activity that you want it to have. Or you run like some DEL screening or some fragment-based screening and such, or maybe you already have like some understanding of which part of the compound, so which substructure is responsible of your activity. And what you want to do now is to find new compound that will have that substructure. But most of the time, you start actually from nothing, so you don't really have anything yet. You start from public data. So if you look at public data like Campbell, PubChem, you have a lot of like people have run experiments on different targets, and they were able to measure like some property or activity for a compound. So can you actually use that to kickstart your drug discovery campaign? So can you build model on this public data to be able to use them to score compound to get a first set of data that you can profile in vitro or in vivo assay to get started on your campaign. So starting from this type of data, you can build a machine learning model. It's not very different from what people have been calling like QSAR, so quantitative structure activity relationship, or like structure property relationship. And this will be on a set of some criteria that you define for your project. So it can be potency, selectivity, some ADMET parameter that I talk about and such. And then when you have those models that have been trained, you can use them as a way to look for new compound that will satisfy the set of criteria that you define. So maybe you have a threshold on potency, so you can use your model to screen a catalog of compound and see which one of these compounds will satisfy the criteria that you define for your projects. Or you can actually use the model that you built to use generative design to kind of design new molecular structure that will satisfy these constraints. And you run through that. So at the end of the presentation, we will talk a bit more in detail about why this is not enough and why it might actually not be working that great. Okay, so we know that we can build machine learning model. We just need some data, and simple as that. So people have been using machine learning in other fields for a while, like computer vision, NLP, on tabular data and such. So in our case, it's pretty much the same thing, right? So we just need some data that's labeled data, where we have some annotation of the molecule telling us whether that molecule is good, or by that I mean active or inactive. And then we put that through our machine learning model that we are selecting, like machine learning algorithm. And then at inference time, so at test time, we have a new molecule, and we don't know anything about that molecule, and we just want to put that molecule through the model and let the model tell us whether that compound is active or not. Quite simple. So why, in reality, it's not that easy? Because first thing is, how do you actually put a molecule in a model? So how do you computationally represent the molecules? And I think I'll talk a bit about some of the representation approach for molecules. I will try to cover them in depth. But also a key issue that we have in drug discovery is that our data sets are very small in general. So in computer vision, you have millions, billions of images and a lot of annotation already available for them. But in a single drug discovery campaign, you usually have, if you're very lucky, 1,000-ish molecules. And you don't really have a full coverage on the parameters that you want to optimize for. So usually you start with a level of coverage on potency, but usually medchem, they don't want to test compounds that do not have good potency. So you don't test them on admets, you don't test them on permeability and such. So you have like a data table with a lot of missing value, and missing value in general is not quite good when you think about like doing optimization on a lot of parameters, because you are reducing the number of like sample that you have on some of the key parameters that you want to optimize for. So that's also another issue. Other thing is that biology is pretty hard in general, and based on that, you have a lot of noise in your readouts, meaning that if you test the same compound several times, you might have some uncertainty on the measurement, so you have some level of noise that is coming from some biological factors, but also like the type of experiment that you're running. It's not only that, like if I say that I'm measuring the binding affinity of a small molecule to a protein, there are hundreds of like different assays that allow me to measure that, but they are not necessarily measuring the same thing, and they are not necessarily fully comparable. So you might have like a lot of data, but it's a lot of data across like different experiments that are not compatible. So how do you bring all of them together, and how do you use all of them? That's also another issue. Other thing is that in general, you would like to have some level of like uncertainty in the prediction that you're making. So your model can be wrong, and if it's wrong, you would like it to provide some level of like uncertainty about like the prediction that it's making. So if a model is wrong and is telling you with full confidence that this is the value, it's not helping you. So you would like to kind of like be able to measure what's the uncertainty on the prediction that the model is making, and that helps you to make better, like more informed decision on how you want to progress your drug discovery campaign. Finally, like there's that notion of like that is called out of distribution. So you will see that a lot where we claim that machine learning models, especially in the context of like drug discovery, they are pretty good in distribution. But when you are moving them to samples that are out of distribution, you lose accuracy. So I will try also to cover what we mean by out of distribution and in distribution. Okay, so let's start with something critical when it comes to like machine learning for drug discovery, which is the representation. So there are a lot of tasks that we need to do in machine learning for drug discovery. First is we need to build a model to be able to score compounds. We need to be able to generate new compounds, so then allow us like to kind of to move away from non-compounds. So if you want to like have a patent on your compound, you need to show that there might be some novelty there compared to what exists already. You also need to like to optimize. So given some predictors, given some generative model, you want to like to bias the generative model into generating compound of interest for you. So also you need some optimization algorithm for that. But on top of all of that, one thing that is critical is the molecular representation. So how do you get a molecule into a machine learning algorithm? And I feel like Bao talked a lot about that, but I really like that this quote here, which is basically saying that however strong is your machine learning algorithm, if you're using the wrong representation, so a representation that is not capturing useful information, you will not get anywhere. And even if you're using the right representation, but you're using the wrong data, you will not get anywhere either. And if even you are using all of that, good model, good representation, good data, but the biological or chemical question that you are trying to ensure is not the right one, is still not useful. So like you need to think about all of them simultaneously. It's not just like throwing machine learning into your data and hoping for the best. It's likely will not work. So what actually is a good molecular representation? So a good molecular representation is one where you can preserve the information contained in the structure of the molecule. So we want to preserve some molecular information. You want to kind of like have a representation that is robust, meaning that if I do small changes in my representation, I will not see like large difference in activity or like prediction of my model, but also like a representation that allow you to build models that are very accurate. Given that we are in the era of like a transformer GNN, so you want a representation that is compatible with this new state-of-the-art like algorithm. And also like very critical is that your representation needs to be relevant to the task that you're trying to predict. By that, I mean that if you decide that my representation is just I'm counting the number of like hydrogen bonds in my molecules, and you use that as a way to maybe predict the, let's say like the metabolic stability of your compound, is very unlikely that it works. So if you have any prior knowledge, any understanding of the chemistry or biology, you will like to like embed that into your representation. So it does not need to be like doing that manually, but making sure that your representation is able to capture some of these like insight is critical to get good performance. Okay, so when we talk about like molecular augmentation for machine learning, there are a lot of like different way where you can represent a molecule. So you can think about fingerprints, test representation of molecules, but also like things like using a molecular graph and such. So each representation has its own strength, but also like some limitation. So not all representation are compatible with all machine learning algorithms, and also not all representation capture like all the information that you need for your task. And in general, like there are kind of like two different way of seeing molecular representation. So either the representation that are handcrafted, so like meaning that there is some expert that look at molecule and say like, this is what matters for me for this task, and versus like a representation that you let the model learn what is meaningful from the molecular structure itself. So let's talk about like the information that is contained in this representation. So I really like this figure here from Connor. So basically you have like a different level of information when you think about a molecule. So you have like the 1D structure, which is quite like a common. So like in this case, like a way to represent that is just saying that I have like a one hot encoding where I put one for my given molecules. And if is another molecule, so at another position, I will put like one there, and all the other entry of my vector will be 0. Then you have the 2D graph, which is a lot more intuitive. That's how we think about molecules. So it's kind of like 2D structure of the molecules. And the 2D graph, you can encode that into a fingerprint. I will go in detail about that, a molecular graph, or you can convert that to an image if you want to, or you can use the 2D graph to extract some handcraft feature like molecular descriptor and such. You can also like convert the 2D graph into like a test representation that we call smiles. Also, between the 2D graph and the 3D information of the molecule, you also have, people tend to call it like 2.5, like D graph. But basically, the idea here is that you are bringing some level of information that is not in the 2D graph, and usually it corresponds to like stereochemistry. So like you, if you are not familiar with that, is kind of like stereochemistry is how you might have like atoms, how they are arranged, like in 2D, they have the same molecular graph, but if you look at them in 3D, it's different. So you might have like some bond that are forward or reverse, and so like it's kind of like a relative position between them. So like being able to capture that is also critical for some tasks. And a good example is there was a drug on the market for like a long time ago, where that drug has some stereoisomer, and one form of the stereoisomer has the right activity, and the other leads to like a lot of like toxicity complications. So like getting the right stereoisomer is also important for the task that you want to produce. And finally, like you have a 3D conformation because molecules live in 3D dimensions, so they are flexible, they move. You have like atoms that moves and like rotation and such. So like having representation that also like capture that is important for some tasks, like predicting binding, for example. Binding is not a static interaction. So both protein and the small molecule will continue like moving a bit, and capturing that full distribution of like movements or like conformational changes between both of them is sometimes really important. Okay, so first let's maybe go in depth about the 2D descriptor and the structural keys. So those are handcraft molecular features. And the goal here is you have your molecules and you want to like to build a vectors that contain the information about your molecule. So a vector that you can feed to your machine learning algorithm. So a simple way to get started is to maybe you want to like to check if there is a specific substructure of interest for you that is present in the molecules. And you have maybe a binary vector where each entry in your vectors correspond to like the presence or the absence of like a specific substructure. So you can put 1 or 0 in each of these entries. So there are examples of that, like the mask key, which is like a 166 like a vector where you check some specific substructure, whether they are present or not. And this was done like several years ago. But also you have like 2D descriptors. So here also like you have a vector where each entry corresponds to like a given property of your molecules. So you might want to count, for example, the number of like hydrogen bond donors, hydrogen bond acceptors, the number of rings in that compound, or you can compute other type of like a PSChem descriptor like the C-log-P or molecular weight, TPSA and such, and you put all of them into a vector that is more or less like describing your molecules. So that's why we say 2D descriptor. And those are kind of like very fast to compute. And depending on your tasks, there can be like a good baseline for you to try to like improve upon. Okay, so outside of this handcraft, there's also like the ECFP, so it's also known as like Morgan fingerprints. And the idea is still similar to checking the presence of like a substructure and putting like 1 or 0, but it's a bit more advanced compared to that. So how it works, you define a cycle around the given atoms. So you loop through all the atoms in your molecules, and then you define a cycle with a given radius around that. And then you will extract like all the substructure where the center is that atom that you picked. So like for maybe like diameter 0 here, and if I'm looking at this specific atom, you have like some set of like substructure, and if you look at like diameter like 2, you have some other set of substructure and such. And then you can pick each of these like substructure, usually we call them like atom environments, and then you can convert them into like you can hash them. So similar to how we hash like a text or image into like like a string or like an identifier. We can do the same thing here, and we'll give you like an integer. And then the goal is that you have a set of like identifiers that correspond to like each of the atom environment for different radius that you are seeing in your molecules, and you can use all of them, and you can fold them into a fixed length vector that will be a binary vector. So that's more or less like a ECFP. And ECFP are pretty much like the go-to molecular representation when you want to get started, you want to test something. Is really like the one that you should be thinking about, maybe trying first before like improving on complexity and such. Okay, so ECFP also used like a lot for when it comes to like molecular search and like try to like to search through a catalog to find molecules that are similar to your carrier of interest. Okay, so let's get a bit more complex compared to this representation. So there's also like the idea of like pharmacophoric features. And here the idea is pretty simple. You kind of make the hypothesis that there are some features in your molecules that are responsible of the interaction with the target. So it can be usually some like electronic features. And what you want to do is to you want to define these features in your molecular graph. So you are replacing the atoms by these features, and you can increase the fuzziness. So like here, look at some feature here. You can change like you have a proxy graph that is a combination of like these feature and how they are linked together. You can be a bit more fuzzy about how you represent that. For example, by merging some cluster. So you can do that. And the idea is that you can then define some geometric features based on how the location, the position of this pharmacophoric point to hash them into a bit vector. So you can use like pairwise occurrence of like a given pair of like pharmacophoric points across a distance. You can use like triplet, quadruplet, and such, and then you can hash them into a bit vector to use for screening and model building. Okay, other stuff, we talked a lot more previously about representing molecules as tests. And when you think about like representing DNA or proteins as tests, is very intuitive. It's just like the list, the sequence of like amino acid or like nucleotides, and that's just more or less like text representation of your DNA or your protein. For molecules, it's not really that simple because these two representations are based on the fact that you can decompose your DNA into a sequence of monomers, so like each of your nucleotides. But if I take like a molecule, like what would be a monomer there? And also like it's not a sequence because you have like rings, you have loops, you have like branching and such. So that's why like SMILE has been proposed and has been used as more or less like the standard on how we represent molecules as tests. And the idea is pretty simple. So yeah, you kind of like you do a tree traversal of your molecules, of your molecular graph, and then you just print out each of the characters that you're seeing. So character correspond to atoms, also like can be bonds. So looking at this here, you can see that we first start by nitrogen atom here, and then we run it through until like we get to like the end of the tree. And every time that we are seeing a branch, like we put that into parentheses. So like here you have this, and when I'm here, there's another branch that is different from the one that I'm following. So I open a parenthesis and I follow through it. And one thing here is that we still want to like to maintain the rings. So like we need to like kind of like track the opening and closing of the rings. Otherwise, we will not have cyclic structure in what we are enumerating. So whenever you have an atom that is part of the ring, you will use digits to know that it's part of this ring here, noted by 1. And when you arrive to like another atom that is part of the same and is closing the ring, you put the same digit. So it gives you some representation like this. And spirals are kind of like a very easy way to represent molecules, both for storing them, but also like we can run like things like transformer on top of like these test representation of molecules. And the nice thing is that it's also like preserve information about the stereochemistry of the molecule with some level of stereochemistry. Okay, so raise your hand if you think that for each molecule there's only one smile string. So based on the description that I kind of like gave for smiles, for any given molecules, is there only one smile string, or do we expect to see a lot of like different way of representing that molecule as a smiles? Yeah, indeed. So a lot, because like rather than starting at this point, I can pick another atom as a way to start. So I will get different like tests that represent the molecule, but also like how I'm kind of like opening and closing the parentheses or the ring closure will give me like a different kind of like test corresponding to a molecule. So in general, for any given molecule, there are a lot of ways to represent them as smiles, and that actually give us like something interesting, because we can use like the random, we call it like smiles randomization. So we can use like that as a way to augment a data set when we are trying to do like molecular modeling. Okay, so smiles have a lot of issues in general, because first there's that issue about like uniqueness. For a given molecule, you have a different way of like representing them as a tests, but also like when you are doing like generative modeling smiles, there's some validity issues with smiles. So people have been proposing different ways of like kind of like representing molecule as tests, and some of them are selfies that kind of ensure that you have validity all the time for your generative modeling, but also things like save that kind of enforce like a fragment-based decomposition of your molecule, which means that you can see all of the fragments being in the same substring, which is not the case for kind of like smiles. Okay, let's talk about 2D molecular graph. So you can see that a graph representation is quite intuitive for molecular structure. So how do we actually represent molecule as a graph? So it's kind of like first thinking about like atoms as node for your molecular graph, and you can build an adjacency matrix that tells you like which atoms are connected. So it's square matrix where you put like 1 when there is a bond between the two nodes. You can also like add some node feature, so some properties of your atom. So it can be the atom type. So is it carbon? Is it like hydrogen and such? You can add other information like the charge of your atom. So is it like positively charged, negatively charged and such, and also like things like the number of implicit atoms and hydrogen and such. So those are kind of like trying to like transfer the molecular graph information into matrices that you can use for your GNN letters. But you can also like try to like encode information about the bond, because all bonds are not the same. You have double bond, triple bond, a single bond. So you can try to encode not only the presence of like a connection between atoms, but also like the type of connection that you have between them, which will be the edge matrix here. Okay, another question. The smiles test-based representation of the 2D graph. Raise your hand if you believe that the graph is better. For any random task, like in general, like let's think about it in terms of like a level of information that is preserved in the smile versus the 2D graph. Graph is better, okay? Yeah, so actually no. So there's exactly more or less the same level of information in the smiles in the 2D graph, if your graph is able to capture the stereochemistry. If it's not able to capture that, then smiles is better. So like graph is actually depending on how you encode it, might be losing information. So, but yeah. Okay, so why do we care that much about having that many representation? The answer is simple. It's what I was saying before, is that different representation encodes different features of your molecules, so give you like a different structure of your chemical space. Meaning that there are representation that makes like some tasks easier than other. And you can see here like there's a bunch of representation that have been mapped to using your map, and then the color of each of the compound here is the solubility of that compound. So you can see that each of the representation will induce a different structure of your chemical space, but also you can see that in some representation, it's a bit easier to kind of like segregate high solubility from low solubility. So in general, when you're trying to build the machine learning model, you want like to search over your representation because some of them might be better for your task and such. Okay, so we know that different representations have different level of information, but also we were talking about not all representation can be used with all machine learning algorithms. So there are kind of like some map between representation and machine learning algorithm that you need to take into account. So for example, if you represent your molecule as test as smiles, it's a bit trickier to try to apply like some linear regression method on that, because first like the length between different molecules is not the same, but also like how do you bring back that test feature into like something that you can use for your linear regression model. Same if you use a smiles representation, well, you cannot really use your state-of-the-art GNN for that. You might be able to try, it might actually work, but like it's not like intuitively the thing that you should think about when you are using that test representation. So thinking about like the representation also like involve like thinking about which type of like machine learning algorithm that I want to use on top of that representation. Okay, so let's take some time to talk quickly about like GNN. So what's actually GNN? So GNN are kind of like a model that are built on some principle that is called like message passing. So the idea is that we can represent our molecule as a set of like adjacency matrix that capture like the connection between nodes, but also things like the node features, but also like the edges features, so like which type of bonds. And then the goal is that we can run like a set of functions that will update the information of the features of each of the nodes in my molecular graph. And at the end, we can kind of like pull all of them together to get a graph level representation. So you start by updating like the node information. So by getting like information from the neighbor of that node. So see it as here, like we have the edges features, and then you have like the node information for the current layer of your graph neural networks. You can get information from all of its neighbor, given the adjacency matrix that you have. And then you have a function that will transform all of this information into a new information that will represent some message that that node is getting from its neighbor. And then you can think about how you want to aggregate that information. You want maybe to sum it over, you want to take the mass, or you want to take the mean, or you want to use things like a PNA where you try to like to learn actually by kind of like going over all the way to pull that message into your nodes to learn like what would be the best way and to get that information, but also like to preserve as much as possible information that you might not otherwise like get if you're only using like one type of like pooling. And then you get that message, and then you have your current node features. You can use that message plus the node feature that you have currently to update through a new function that will give you like the new node representation at the next time step. So you can do that several times. So you get message, you exchange message, and then at the end you can pull all of that into a single vector. So right now we still have like a vector per node, but we can do a global pooling and put all of that information into a single vector that correspond to the graph representation, and then feed that through a MLP to kind of predict for a given task. Similar, we can also do something with tests. So rather than using GNNs here, we can use something like a transformer, where the idea is that we start by the test representation of the molecules. And then in contrast to like how we convert like our tests in NLP to tokens, for molecules, we need to do it in a slightly different way to be a bit more optimal. So you can still like kind of like tokenize your molecular tests by character, but like empirically, if you are able to like to leverage like a better way of doing that, for example, either by using kind of like by being aware of like the functional group or like the substructure into your inside your test, or even just like by preserving it by doing the tokenization in such a way that you preserve the atom identity, it can improve like performance. Because remember that in the SMILE string we have characters that are not atoms, that are not bound, like the parentheses of a digit, for example. So making sure that we have a tokenization that is able to capture things that are more chemically relevant can help. So you have that, you pass it through your embedding layers, and then you can use like the usual transformer as a way to like to try to predict some learn a model to predict some given task and such. Okay, so I feel like maybe the most useful thing that I can maybe share with you today is that idea of like it doesn't really make sense when you have a molecular data set and then you start throwing your state-of-the-art like algorithm to that. First, try to think about like what is actually the task that I want to accomplish. Then think about like what would be a strong baseline for that task. And you will see that we usually say that as a joke, but like random forest and ECFP is very strong baseline when it comes to like chemical modeling. And it's true, like even if you do beat them, it's barely by some small margin. So like first start by building a baseline before trying to like to build like your state-of-the-art or large model and such. Okay, so a few things that we might also need to cover is that in molecular modeling, we have some issue that is called activity cliffs, and the principle is pretty simple. So you have two compounds or several compounds that have very similar structure, but they have very different activities. So meaning that at least when you look at the structure, you will expect them to be similar in terms of like activity, but it's not the case. So like, how do we build like machine learning models that are able to like to capture like the simple changes that you are seeing in the structure and the effect on the prediction? And this comes down to like mainly to the type of representation that you're using. If you're using a representation that is invariant to the effect of these changes on the activity prediction, then you will be able to have good performance. But if your representation is very sensible to that, as in you see them as close, but in the target distribution they are not close, then you will struggle to try to predict like the activity. Same for like that concept of like outer distribution in molecular modeling. So what we see is that usually when you are trying to test your model, as you are testing on a set of compounds that are a bit further away from your training set, you will see a decrease in performance. And that's usually because like what you are testing it on is not really what you have trained the model on. So the model will struggle to have good performance in those new distribution. And this is what we call out of distribution, because it's not the same distribution as the one that you're seeing in the training set. And a good, kind of a simple way to tackle that is by that idea of like applicability domain. So basically, you try to set some chemical space around your model that you will be able to use as a way to say that within this space, my model will perform better and will be a bit more robust. So it's kind of a very naive way of like kind of trying to address OOD. by kind of like setting some constraint about where the model will be the best in terms of like performance. But one issue is that how do you define applicability domain? So like, do you just say that if my compounds are too far from my training set, given some threshold of distance, then they are OOD, and then I should not predict on them. No, so like the right way is actually to be able to assess the uncertainty in prediction that your model is doing. And maybe like a good way to try that is, I have these 4 molecules. Raise your hand if you believe that molecule 1 and 4 are the closest. Okay, so we have 1 person that believes that 1 and 4 are the closest. Any other choice? Like they are like, it's only the like yellow cycle that there are some differences there. So you see that it's actually hard to evaluate similarity from a human perspective. And if you run all of them through like ECFP, and then you compute the Tanimoto distance between all pair, you see that molecule 2 is actually closer to molecule 1 compared to the other. But intuitively, you see that there are a lot more changes in the molecular structure between 1 and 2. So like that idea of like defining a distance to be able to say that my model is within the applicability domain. It seems simple, but it's kind of like, it's hard to get right. Okay, so in general, if you want to like to address like OOD when it comes to like to molecular modeling, a good way to get started is to think about how you are splitting your data. So like if you have a training data set, how do you split your data in such a way that you are trying to maximize the impact of your model when it comes to like inference time or deployment. So you like probably to avoid the random split, because there's a high chance that there will be like some leakage between the training set and the set that you will be evaluating your molecule on, your model on, sorry. And there has been like some proposal like doing a scaffold split, which is a bit harder, where you have like some set of scaffold that are not in your training set. So if your model is able to have good prediction on this test set, that means that it's able to like generalize a bit better to a slightly different chemical space. If you have like a time information on your data set, you can also do a time-based split, which basically is more kind of like match a bit more how we do drug discovery, because we progress like compound through times. So if you're able to use a compound that were at the first stage to predict the property of like compound at the later stage, then you have a good potential of like generalizability. And in this paper, we were actually proposing a way to actually prescribe which type of split you need to do to ensure that your model is able to generalize better when it comes to like to the deployment sets. Okay, I have maybe like only 1 minute left, but I think I will try to like to kind of cover quickly that idea of transferring knowledge, which is very important when it comes to like machine learning for drug discovery. So like we know that we don't have enough data is noisy and such. So can we use like other type of like information data to kind of like try to improve the prediction of our model? And you can do that by leveraging like existing bioassays on maybe related tasks, or you can also do that by trying to learn from data where you don't have any label. So that brings us to like some of the concepts about self-supervised. So maybe you can train a model to learn more about chemistry by trying to predict part of your molecules, or by trying to predict some given basic molecular properties, and that allow you to have like a representation that's a bit more robust, that capture more about the chemical information that you can then transfer into your task of interest. You can do the same thing more or less with like a multitask learning, where you have a data set and you try to like to predict on a very massive like set of like tasks. And the goal is that by predicting on all of these tasks simultaneously, that you are learning there might be some synergy between the tasks, or let's say like solubility and permeability. So like by predicting them like simultaneously, you are capturing like more common knowledge, more common chemical information across the task that will be useful for later downstream task. So there has been like a lot of like interesting model that have been pre-trained on like similar tasks, like either we are doing self-supervised or we are doing like a multitask, and then after training we are able to like extract then like the features from our large model and transfer that to like a new task. And this kind of like pre-trained molecular representation are kind of like where the field is going right now. So like doing that in a very large scale allows us like to build like representation that are able to capture more about the chemistry and more about the biology. And those are representations that are learned from the data itself, which is pretty useful. So there's kind of like a list of like these state-of-the-art representation, pre-trained molecular representation in GitHub that I will invite you to check. Okay, so finally, like let's end with like active learning. So the goal of active learning is we know that drug discovery is expensive. So testing like just testing like permeability on CACO2 is like more than 500 like a compound. So you cannot do that like if you do that for 10,000 compounds, I don't think that your startup will be that successful. So you need to be like very smart about the way where you profile compound. And this is kind of like the idea of active learning, where basically at each step of profiling, you will select compound that will bring the most information to your model, so that will allow your model to be able to generalize better and such. So rather than selecting compound that will maximize your property of interest, you want to select compound where when you get the feedback, the profiling results from them, you will improve the model. And this is the main idea of like active learning. Then how you do that is you need to define an acquisition function. So a function that will tell you that these are the molecules that will bring the most information to my model, and that is usually based on uncertainty. So you need a model that is able to predict some uncertainty, and by that, it means that the model, the algorithm itself needs to support that. And there are a few of them, like you can do like Bayesian based inference. You can do like use assembling. So you have like set of model, then you take the average as a way to get like the prediction, and then you take the variance of the prediction as a way to get some level of uncertainty. And there are kind of like other class of like algorithm like MVE, like MC dropout and such, when you're using neural networks that allow you to provide that level of uncertainty, and then you can use that as a way to like acquire new compound, profile them, and improve your model over time. Okay, so finally, like we are getting back to that slide that I showed about you have your model, you can use it for virtual screening, but you know already, like you don't need anyone to tell you that it will not be a good model. So from the start, you need to be in a mindset, you need to be like in a setup where you are testing compound with a specific purpose, which is to improve a model for the next cycle. So like, you will see that a lot of people in this space talk a lot about active learning because we know that it works, and it's kind of like one of the only tools that we have to deal with the very low data that we have in general in drug discovery and all the noise and such. So if I improve in the certainty of the model, the performance of the model over time, you are able to like to have a better impact on drug discovery in general. Okay, so I think that's the end of the presentation. And in lab 1 and 2, you will cover part of the material about molecular computation, about building machine learning model, about like active learning too.
We have time for 2 questions. So if you have a question, please raise your hand. I'll bring the mic to you. Okay, let's start here.
Thanks for the talk. How many active learning loops are there operating right now on planet Earth successfully?
Yeah, that's a tricky question. I would say that it depends on your program. So if you are getting started with a model that has some level of performance already, you might not need to like do a lot of cycle before getting to a model that generalize in a good way. But it also like depends on the training set that you're starting with. So if you're only starting with a few data points, it's more challenging to get to like model that generalize. But also like it depends on the budget that you have on each cycle. So how many compounds can you test in each cycle? So you can understand that if you are testing more compound, you are bringing in more gaining information to the model. But if every cycle you're only testing like 5 compounds, that might be a bit more challenging.
My question is also related to active learning. You said that it works. So I'm actually curious, if you have multiple query strategies, how would you compare which of them is superior? And yeah, technically, how would you compare that it works better than not active learning?
Yeah, so that's also a very good question. One of the challenge when it comes to like active learning is that you are locked on a single model. So like you cannot do active learning on several models at the same time because you need to pick the one, you need to improve only one by like gaining new information. But also like you need to be careful about the acquisition function that you want to test. And that will also depends on the type of architecture that you have used to build your model. So in some cases, you might think about like using some acquisition function that depends on like expected improvement, for example. But in other cases, maybe it's just simpler to maximize the diversity of your pool of candidates. So when you are uncertain about how to actually pick the molecules, a good strategy is to profile as many diverse compounds as possible, because diversity usually doesn't hurt in improving the model.
Oh, hello? Okay, cool. Hi, thank you for your presentation. I just had a quick question on the computational efficiency when it comes to 2D molecular graphs. I know you mentioned like when it comes to stereochemistry, they carry like just a little bit more information in comparison to like SMILES. But how will it affect, I guess, like your model runtime with using those potentially large matrices? Thank you.
Yeah, so that's also like a good question, and I think like part of the answer is about like developing tools that will improve the efficiency of GNN in general. So like when it comes to things like transformer on test, people have been working on that for a while, pretty excited. There have been like a lot of engineering effort to make them efficient. So you have things like flash attention for transformer and such. You have whole package like the transformer from Hugging Face that allow like people to get access to that quickly. In the GNN community, it's not necessarily the same. So like you have a few things like PyTorch, geometric, you have Graphium, and also like the DeepChem library. So there's still maybe like a lot of engineering effort that needs to be done there to like improve the efficiency to make like inference like cheaper and faster.
One last question for myself. How unsupervised and pre-training help with the domain of applicability of the model?
You're referring to?
So basically, how can
Yes.
you use? Yeah.
So how can we use
pre-training and unsupervised
yeah, so like
learning?
Yeah, so that's also a good question. So first, the issue with applicability domain is people tend to define them on feature set that is different from what they have been using for training the model. So you see people define applicability domains by looking at tiny motor distance between the molecules on ECFP, but they had trained their model using a GNN, for example. So they have it's different perspective. So it's different representation of molecule. So by moving to pre-trained representation in general, we are not only trying to standardize the in the molecule, but also potentially the representation that we are using to define the molecular domain, so the applicability domain of the molecule. So which will kind of simplify a lot, assessing whether the molecule is within the distribution of your training set or not.
Perfect. Let's thank the speaker once again.